So thanks everyone for coming today. My name is Chloé Charendoff. I'm one of the field application specialists for the Rhapsody system. And it's my sales counterpart, Katie Garrett. And today I'm going to give a bit of a portfolio overview of the different assays and options you have to using our Rhapsody system for single cell multiomic experiments. So I wanted to start by considering, why is single cell RNA sequencing such a powerful tool? So here we have this study in which the investigators wanted to characterize 29 different subpopulations of immune cells. And so what we're looking at here is the number of unique genes they found in these different subpopulations versus transcripts per million. And this is a normalization technique that allows for easier comparison of the proportion of sequencing reads mapping to a given gene across samples. So what we can see from both of these figures is that across different cell types, we see quite a range of gene expression and heterogeneity. So we want to use tools that can provide finer resolution so that we can see what are the cells in these different subpopulations doing, how are they interacting within a subpopulation, and how might they be interacting in a heterogeneous landscape under the context of disease, for example. So a tool you might be familiar with from BD is going to be probably our cytometers. So we have a lot of flow cytometers and a lot of history in that space. And it's a great tool for real-time analysis at a single cell level, where you can look at over 50 parameters at a time, depending on which cytometer you're using. However, with single cell technology, we can actually assess thousands of parameters per cell on a single cell basis using our Rhapsody system, and that's what I'll be talking about today. Throughout the presentation, I'm going to touch on some examples from the literature in which some of our users have used our system to publish their own single cell data. So hopefully you find some fun inspiration from these publications. So this one in particular is a favorite of mine. In this particular publication, the group of investigators were interested in determining the differences in immune cell composition and activation in patients with mild COVID-19 symptoms versus severe symptoms. In doing so, they became interested in the myeloid compartments. So they found that effectively, neutrophils, when patients that had severe COVID-19 symptoms, undergo this emergency myelopoiesis, which results in immature and dysfunctional neutrophils and could be connected to some of these symptoms that they see. So that's just one example of many that I'm going to talk about of a group that used our system to generate some really cool data and a really cool story for publication. So our portfolio supports a wide array of analytes, and we support the instruments through reagents, through some data analysis, and we'll talk about that as well. We can analyze multiple analyte types from DNA to RNA to protein, and a lot of these we can do at the same time. And this, of course, supports many different research areas. It's a very flexible tool to use and can answer questions in multiple different subdisciplines. So I wanted to give an overview of what a Rhapsody experiment looks like in its entirety from start to finish. Many of our users are interested in a rare subpopulation, so they may choose to do some kind of enrichment. This can be flow cytometry, this can be like a magnetic selection column, but something to enrich for your cell type of interest. It is, of course, optional. We also have plenty of groups that are interested in a more heterogeneous mix of cells. From there, we may move on to our SCIC assay, which I'll go into more detail about how that works, but this is what we use to look at protein expression. They may do multiplexing or cell hashing. If they're interested in the TAC or TAC multiome, they may do their tagmentation steps here, but all of this is part of sample processing. From there, we move into cell capture on our instruments, which I'll go into more detail here momentarily. And that is where we partition cells into single wells of our capture cartridge so that we can isolate them with one of our cell capture beads and hybridize that genomic and proteomic information to our capture oligos. And this is our first stable stopping point in the Rhapsody experiment. We do generate cDNA stably on our beads, and it is stable at 4 degrees for up to a year. So it's a really nice and flexible part of the system that allows you some time to bank samples or send them to collaborators or things like that between your live cell capture and your library preparation. After that, we use those beads that we've generated our cDNA template to generate libraries, be that for your RNA sequencing, for full-length VDJ, for those that are interested in T-cell and B-cell repertoires, ATAC-seq, whole transcriptome, kind of all of our various assays, you would generate libraries for them here. And then our libraries are ready for sequencing. These are generated for Illumina sequencing. We do, however, have compatibility with Element, and we are also currently working with Ultima for those who are shopping around for different sequencers. And then we have data analysis and interpretation, which I'll also touch on today. We offer free to our users a primary analysis pipeline, which is hosted on 7bridges, as well as our secondary analysis data visualization tool, the Ucilismo, which is a point-and-click user face for our end users who don't have experience or are not proficient coding. It allows you to do some data exploration while you wait for bioinformatician to really dig into it. So really, we have our live cell workflow where we're working with live cells and our library preparation and analysis. So I want to get into the instrument itself, the different workflow components and how they work. So these are the basic workflow components for our capture. We have our HT Express. This is our cartridge loading station and the workhorse of our instrumentation. This is where we load our high throughput cartridge. Anytime we're putting something into the cartridge, it'll sit here on this instrument. We load this cartridge with an automated pipette. This is a P1000 pipette. It's automated for every step of the workflow, and it's programmed for both volume and flow rate at each of these steps. So it's very gentle on your samples, and there are no tricks to loading the system. There are no microfluidics in the system, so no samples are going to get clogged or lost. So it's very, very easy to use. We then, of course, have our cell capture beads. These are paramagnetic beads that are coated in our capture oligos. And this is what we're going to use to capture all of those analytes that you're interested in. And then we have our scanner instrument. This is our instrument that we use for quality control, and I'll go into more detail about some of the metrics we get from the scanner. But we can use it as a cell counter, and it also is going to give us information about how many cells have been captured during the experiment, which allows us to better calculate for our sequencing needs. So taking a closer look at the HT Express instrument, as I mentioned earlier, this instrument has no plugs or microfluidics. It's actually pretty small and easy to manipulate and move around. It only has two moving parts, this front slider here and this magnet up top. So it's super, super easy to use. It's easy to move as needed. So if anyone's working on something potentially infectious, you can put it in a biosafety cabinet very easily. And there's no risk of losing your sample due to clogging or wetting errors. And then we've got our high throughput cartridge. So this is our where we load our cells for partitioning. It has 8 lanes, and each of these lanes has over 267,000 microwells in it. And we can load between 100 to 100,000 cells or nuclei per lane. It is flexible by design, so you do not have to use all 8 lanes at once. So this is a really nice feature of our system. So you can use all 8 lanes, you can use 1 lane, you can use 4, however many you want. And when you're done, there's a cleanout procedure that helps us to dry the used lanes, and we store it in a resealable pouch with a desiccant package. Once you open your cartridge, you have a year to finish using it. So this is really nice for groups that maybe get a sample at a time and don't want to have to buy a new chip or cartridge every time they run a sample. As I mentioned, we have a dynamic range of cell inputs, so as few as 100 cells and as many as 100,000 cells, with a multiplet rate of about 0.2% per 1,000 cells. So very low multiplet rate compared to some other platforms. So this is just a quick overview of what's going on in the wells during that partitioning. So we load our cells into the system and we allow it to settle very gently into the wells via gravity. This is a really nice system for fragile cell types like granulocytes, as we're not putting them through any stress when we put them into the system, and they just use gravity to settle gently into those wells. Then we load those magnetic capture beads and allow them to settle into the wells. And then we lyse those cells and allow anything polyadenylated or tegmented to be to hybridize to those oligos that are on our bead. And then we engage that magnet in the top of the express to pull the beads up out of the wells and flush them into a tube for cDNA synthesis. So I wanted to highlight some examples from the literature for groups that have struggled using droplet-based platforms to get single cell data. This group in particular comes from Carl Fernandez's lab at Sherbrooke. And here they were interested in looking at the role of microglia in their Alzheimer's mouse model. And they had struggled previously to capture these cell types on the 10x system. I won't read the whole thing to you, and I'm happy to send these slides out when we're done. But just wanted to demonstrate that using our system, they were able to capture these cells effectively and look at their role in this model with this particular desaturase they were interested in. Similarly, we have this group out of, I believe, Austria that was trying to make a single cell atlas for their non-small cell lung cancer model, and they were struggling to find their granulocytes in physiologically relevant proportions using the 10x platform, and found that not only were they able to find these cells using our system, but that they were able to capture a notably higher number of mRNA molecules per cell. So really great system for all cell types, especially fragile cell types that are prone to lysing when you look at them a little bit funny. So now I want to talk about our scanner and what we use it for. So this instrument is really great I really like it for my new users. I really like it for anyone that maybe is a little bit nervous about running a single cell assay, because this is our quality control instrument. So this does have a cell counting function that we can use to determine the viability of our samples. It will calculate for us how to make a pool of our samples and give us a predicted multiplet rate. And this is really nice in and of itself, as it gives you a really nice go, no-go decision-making option for whether you're going to run your samples that day. It also gives us information at every step of the capture workflow regarding how many cells have been captured, did we load our beads properly, how many wells have a cell and a bead, as well as our retrieval efficiency. So I showed you guys this slide just moments ago where we talked about the steps of the capture workflow. And effectively, we have a scan for each of these steps. And at the end, we have a really nice metrics report for the end user to indicate a whole bunch of metrics, but most notably this one here, which is the number of wells that have viable cells and a bead. So this is kind of your money number. This is going to tell you how much sequencing total you need for that experiment. And it will also give you an idea of how many cycles you need to adjust your library preparation reactions. So really helpful here. Kind of rounding out the instrument side of things, why are microwells really effective for capturing single cells? So we talked about the simplicity of the system, only 2 moving parts with that instrument. The flexibility of our cartridge, you don't need to use all 8 lanes at once, so your reagents go farther for these experiments. We have very high throughput and high capture efficiencies for various cell types, and the gentle nature of how the cells settle help us to maintain that sample integrity. And our scanner is really great for that in assay QC, and it allows us to feel reassured that the experiment is going well, or if it's not going well, it allows us to kind of cut our losses and come back another day to try again without wasting money on sequencing. So now I'm going to move into our assays and reagents. We have a lot of different options, and most of them are stackable, which means you can perform multiple assays on a single set of cells. So first, I'm going to go over our options for mRNA sequencing. This is the bread and butter and basis of most single cell platforms. So I want to make sure that we talk about this first. So we have 2 different options for looking at mRNA expression on our system. We have our whole transcriptome analysis, which is where we take an unbiased approach to assay all poly-A transcripts in the cell at the time of lysis. I really love this assay for my groups that are interested in discovery, because we're casting a really broad net here. We can capture multiple transcript variants that maybe you didn't know were relevant to your condition of interest. The only caveat with this assay is that if it's a gene, if you have a gene of interest that has very low expression, we have to make sure we sequence deeply enough to find it in your sequencing data. And this is generated via random priming and extension. We also have our targeted transcript amplification approach. So here, this is a great assay in that it is very sensitive. So here we're using transcript-specific primers to target genes of interest. Because we're only targeting genes that we care about, This is a very high resolution and sensitive assay. However, we will not find what you are not looking for. So you do need to know ahead of time your genes of interest here. This is a really popular assay with my groups that have done a lot of single cell work, and they're looking to verify a transcriptomic profile that they already know very well. It does require fewer sequencing reads than WTA, so it often ends up being cheaper to sequence. So it really is project dependent, which of these best suits your needs. And just another way to look at whole transcriptome versus targeted, just to kind of help visualize the differences. With whole transcriptome, we like to think that we're looking at the big picture. And with our targeted assay, we're looking at a smaller picture, but the resolution is more fine. For targeted option, we actually have a lot of options. We have some pre-designed panels off the shelf. These are about 300 to 500 genes, depending on which one you choose. If you like the look of these assays, but maybe there's a few pathways that are missing that are of interest to you, we can design a custom supplement for you to add into that backbone panel. Or if you're interested in taking a targeted approach, but none of these off-the-shelf options work for you, we can design a completely custom panel for you. I also wanted to mention that we just released our newest iteration of our whole transcriptome assay, which is called WTA Next. We're really excited about it. We have a lot higher sensitivity and specificity in this assay. We're getting greater than 3,000 genes per cell, which is pretty exciting. It is compatible with basically all of our other assays. So that's going to be ATAC, VDJ, any of our protein assays will be compatible with this new iteration. And it is also compatible with some of our preservation buffers that I'll talk about later, which allow you to store your cells at 4 degrees for up to 72 hours. And it also comes at a lower cost per sample on the reagent cost side. So we're very excited about this assay and happy to talk about it in more detail with you guys after the seminar. Okay, next up, I want to talk about our ATAC-seq options. We're also very excited to include ATAC-seq in our portfolio as it's been very popular lately. So for those not familiar with ATAC-seq, the purpose of this assay is to cut chromatin between histone complexes and find which transcription factors or genes are located in these open reading frames or open chromatin regions. For this assay, we can load up to 1,000 nuclei per lane, and we have options for multiplexing, which means we can tag nuclei from individual samples and put them in one run together, lowering your reagent costs. We also offer our ATAC multiome assay, which is ATAC-seq with mRNA expression at the same time. And I think this is one of my favorite analogies that we've come up with to discuss why would we want to look at ATAC-seq and RNA-seq at the same time. So if we think of a map, right, or like Google Maps, ATAC-seq is going to give you an idea of where all of your accessible roads are. So where are all of your open chromatin spots or positions. Adding that RNA-seq element is more like real-time traffic data. So we know which roads are open, but which ones are busy, which genes are actively being upregulated under your certain conditions. So this can be a really powerful way to look at your samples and gain more understanding of that biology. This is an example of a type of experiment one might do for ATAC multiome, and this actually was a study that was generated in-house, but we do have a webinar where they go over what they did. If you're interested in that, we can send you that link. But effectively, the investigators had unstimulated versus stimulated T cells, and they sorted these on our S8, Discover S8, and performed nuclear isolation that they then brought into our Rhapsody system for ATAC multiome, and were able to generate ATAC data for their open chromatin regions, as well as the corresponding mRNA data that gives them an idea of, okay, of these open chromatin regions, which one of these regions were actually being upregulated in response to this stimulus. Next, I'll touch on our TCR-BCR Next assay. So this is our assay that we offer for looking at T cell and B cell receptor sequencing. We do offer full-length VDJ, where we capture CR1, 2, and 3 regions, and this assay is compatible with our mRNA assays as well as our protein assay and multiplexing, and it allows us to look at these different sequences and give you clonotyping information and really cool stuff for those that are interested in T cell or B cell biology. Our current version of that assay is called TCR-BCR Next, in which we have improved our pairing efficiency and mapping, and also, yeah, is compatible with all of our other assays with the exception of the SCIC. Okay, protein profiling. So hopefully we have some folks in here who are interested in protein profiling. So we've got a lot of cool stuff we can do with it. So in trying to further resolve cellular heterogeneity, I like to bring us back to this idea of mRNA versus protein expression. And we know that mRNA and protein expression do not always correlate. So this is a really nice example of that. When you look at the mRNA expression of this group of immune cells, of CD4, it's not easy to tell which one of these clusters is associated with your CD4 positive T cells. But when we look at protein expression, they light right up, so it's way easier to see, right? So CD4 protein has a much longer half-life than the mRNA transcript. And this is why it can be important to have both sets of this data. Another example here, we have an RNA-driven T-SNE clustering plot where we're looking at various immune cell subtypes. And we get some pretty decent clustering of cell types clustering with like cell types to give us an idea of what these cell populations are. But if we look at this cluster down here, this is actually a mix of our memory CD4s, naive CD4s, and some CD8s. When we layer in our protein information, we're able to get much better separation of these different clusters and subcell subpopulations, which allows us to do even deeper investigations of our data sets. So I wanted to give an example from the literature that is super fun. So this is a group out of Germany. This is Simon Haas's lab, and they made a proteogenomic reference map of hematopoietic stem cells in their intermediate. So their goal in this publication was to close the gap between clinical flow diagnostic panels and the expected RNA expression. So they used almost 200 of our SCIC antibodies in conjunction with our whole transcriptome assay to generate these proteogenomic reference maps and make this map that they then use as a control to overlay a cohort of AML patient data. And what they were able to generate some really cool data from this to include new and revised clinical flow panels that could better characterize different subsets of AML, which was super, super cool. You don't have to use 200 of these antibodies. I just like to show this paper because they really went for it, and it shows really how much information you can generate from these experiments. It also shows how your multiomic experiments, your multiomic single cell experiments can feed into other types of experiments. In this particular example, they were using single cell to better inform their flow experiments. But how do these work, right? So our proteomics assay is an antibody-based assay. We use the same antibodies that we use for BD flow, but instead of a fluorophore, we have an oligoconjugate. And this conjugate has a bunch of barcoding on it to include a PCR handle that we use for amplification. We also have an antibody-specific barcode, so this is what's going to tell us in your sequencing data which antibody was bound to that cell, and then a poly-A tail. So our system is a 3' capture system, so anything polyadenylated in or on the cell at the time of lysis will bind to the poly-T on our beads, and this is how we're able to look at mRNA data as well as protein data at the same time. We have a big, big repertoire of these SCIC antibodies. We have over 458 clones available. About half of them are against human targets and the other half against mouse. We do make custom antibodies, and it does not have to be a BD antibody. So if you look through that repertoire of ours and you don't find what you're looking for, we can make you a custom antibody. We also have some off-the-shelf panels. These are Omics 1 panels. So currently, these are for human immune cells. These are pre-made panels of 30 antibodies or 30 targets that target various aspects of the human immune response. So we have our immune profiler panel, which is a bit more general. We also have our T and B cell panel, a tumor microenvironment panel, NK and antigen presenting cell panel. What's nice about these assays is it can save you money if you're interested in our SCIC options but don't want to buy all of the individual antibodies. You can get one of these panels that come with 2 tests. You can also stack these, so if you wanted, you could put all 5 of these or all 6 of them in one big panel and do 150 targets all at once. So I really love it for folks that are interested in getting into proteomics with single cell, but want to have some kind of introductory pre-made panel to play with. We also have introduced our SmartSeq technology with these panels. Effectively, what we've done is for primary markers or lineage markers, we introduce what we call cold antibodies. These are antibodies that do not have an oligo. And it does a bit of signal muting for some of these really highly expressed lineage markers. And it allows more of your sequencing reads to be allocated to some of your more rare or lower expressed markers. Thus, you don't need to sequence this assay as deeply as other SCIC assays. We also have a really nice SCIC application or assays in that the way our system works, it is an open well system, right? So when we do SCIC work, these antibodies will bind to surface markers on live cells, but they'll also bind to dead cells and debris. You also, if you didn't wash very well, you may have some ambient antibody floating around. What's nice about our open well system is that we incorporate extra on-cartridge washes for our SCIC protocol, and it allows us to remove unbound antibody as well as ambient mRNA and debris, and this is something that's not possible in a droplet-based system. And what this does for us is gives us a higher dynamic range for that protein expression in our data sets with a lower background. So we have really, really satisfied end users that get into SCIC with our antibodies with our open well system. We also have sample multiplexing kits. So while we're talking about our antibody-based assays, this is very similar in concept to how our AbSeq antibodies work, except in one of these kits, you'll have multiple aliquots of the same antibody, but each antibody has a different tag. So the idea here is you can label cells from different samples and assign them a sample number that you can then put multiple samples in one lane of your cartridge. And so this helps with increasing throughput per lane because you can put multiple samples in one lane. It lowers your reagent costs because you're only using one lane of reagents to generate that data. And it can also help with reducing batch effects and allows us to identify and remove multiplex from the data. We have a lot of different options for sample multiplexing, just kind of depending on what your project is, and we're happy to help you figure out which one works best for you. We have one for human samples as well as mouse. We have a nucleoporin tag, which is really popular with our ATAC-seq users, as well as this flex kit, which is species agnostic, and we can talk about how that works if you're interested. Right, we're almost there, guys. So we also have a protocol that we recently published for generating full-length cDNA for those that are interested in long-read sequencing. So those interested in particular isoforms are typically interested in long-read sequencing. We developed our protocol in collaboration with PacBio. We also have the ability to sequence with Oxford Nanopore. And then I wanted to round it out talking about our sample preservation reagent options. So we have our first sample preservation reagent option, which is called Omics Guard. So this buffer is a one-step preservation protocol where we can preserve tissue, single-cell suspension, or whole blood at 72 degrees, or sorry, at 4 degrees for up to 72 hours. So I really like this for my groups that are working with clinical samples or maybe doing a lot of animal work and you're processing multiple animals that you need to run for one experiment, because it allows you to effectively press pause on mRNA transcription and protein generation and degradation to kind of collect everything and then move on with your single cell experiment. It is not a fixed in. This does not have PFA or methanol, ethanol in it. This is a preservation buffer, so your cells are still alive at this time. It comes in 2 different configurations. You can get a 50 ml bottle or this 12 pack of 1 ml aliquots. And in our internal testing, we've seen no to minimal impact on ATAC sensitivity, WTA sensitivity, or our AbSeq assays. So this has been really great for some of my groups who, like I said, have these clinical samples or a ton of mouse samples that they need to process, and it allows us to do your processing day 1 and maybe come in and do your partitioning day 2 and break up the workflow a bit so that no one is in the lab until 1 in the morning. We've also recently launched our long-term sample storage solution, which we call OmicsGuard Cryo. This has been validated for single-cell suspension, and effectively, we add this preservation reagent to the sample, and you're able to store your samples at -80 for up to 2 months, or in liquid nitrogen for 9 months. So very excited about this long-term solution, and it also will be helpful in helping maintain your sample integrity. All right, last but not least, I want to touch on our bioinformatics support and solutions. We do have bioinformatics specialists on our team who can help you navigate these particular platforms. And that is a whole other seminar that we'll let them give at another time, but I do want to mention that we have them. So we offer our primary analysis platform on 7 bridges. All of the assays can be run on the same pipeline at the same time, so that's pretty nice. For those that are familiar with 10x Genomics, it has a similar function to Cell Ranger. So it's here to do some initial processing of your data, some initial QC, and it provides outputs that you can then use for secondary analysis, like Seurat or Python. And it also produces a file that's called a .celismo file, which can be plugged into our secondary analysis data visualization tool, which I mentioned at the beginning of the talk. So this is our point-and-click data visualization tool that allows those of us, myself included, with limited coding experience to do some exploration of the data. Both of these platforms, 7 Bridges and Celismo, are free for our users to download and use and run. And so Celismo in particular has a bunch of demo data sets preloaded on it, so you're more than welcome to get on there and check it out. We in fact encourage it. And our bioinformatics specialists, when you guys are running your samples or are doing your projects, are here to help you navigate these platforms. Insofar as technical support, we do have a website where all of our protocols are housed, but really we encourage you to reach out to your applications team, which for you guys is me. I cover the Midwest, and I'm here to discuss experimental design, any kind of troubleshooting, training. If you want me to QC anything with you, I'm your girl, and I'm happy and excited to work with you guys. Basically, rounding it out, if Katie wanted to come up, we've got a special offer going on right now that she's going to tell you guys about.
Everyone, Katie, I'm your sales rep, so obviously Chloé will be your tech. But we work together as a team, right? So we always want to meet with both of us. But so what we're thinking and working with the core here is hopefully bringing a demo instrument in. So if there are people who are interested and have maybe some samples ready to go, pilot project that you're thinking of for grant submission, we would love to be able to help you out with that. So again, this is just kind of a quick little option. I will, if you have registered, I will be sending this information to you. But we will be here for a couple hours after this meeting if you want to come and have a chat with us. Even if it's just to ask some questions, no more than, just to even go over a project and an idea, maybe not even right now, but something in the future, and we can always talk about that as well. But yeah, so really I think what it's going to try to do is, hopefully maybe sometime in February, maybe about 30 to 45 days, we can get some projects going. We do a training session, and then you guys would be on your own to kind of run your pilot, but we would be here again for questions to help you out any way we can. So I think that's kind of it. Mm-hmm. So yeah, thank you so much. And I'll let you answer any questions that people have.
Yeah, absolutely. So thank you again for coming. We're so glad that you were able to make it. And like Katie said, I'm happy to answer questions now, but we'll also be hanging out over here for a couple hours if you want to come back and chat about the project.
Yeah, and we also have a bunch of brochures, so too, if there's a particular assay you're interested in, we have some things you can kind of look at, like some of those Omics 1 panels, a little card for like the Celismo, if you do want to play with some data sets. So yeah, please feel free to, kind of chat with us and get away about it. So. And lots of food, so it's more sandwich.
I was just wondering, are some of your products available for clinical diagnostics or?
Currently, we are research use only.
All of the company's products?
All of the single cell products.
For the plant preservation,
Mhm.
are the cells still alive?
Yes. The
So you
cells
can
are still
still
alive.
like we digest our samples,
Mhm.
preserve.
Mhm.
Then can we do the sorting later on?
Yes, so that has been, that particular preservation reagent has been validated for single cell experiments, but also flow and qPCR. Yeah.
What's the difference between the syntax group and the neural system and all the cells is separated in the cartridge in the single cells?
Yeah, what's the difference? So,
Yeah, they do when you prepare your single cell solution.
yeah, that's a really good question. So the major difference between our partitioning technology and 10x is our open well system. So we make a pool of cells within a particular concentration and we flow them over our cartridge and let them settle into the wells. And then we add that capture bead that has our oligos on it. So with 10x, they're running it through microfluidics to encapsulate them in their gems. So that's the major difference. We don't have microfluidics in our system. It's an open well system. It is very similar, and I think, in fact, we share the IP for the barcoding on the beads. So our poly A capture is very similar to theirs at the chemistry level. It is the cell partitioning itself that's different. As far as considerations for sample preparation, our loading volume is 320 microliter, which is quite a bit more than the 10x loading volume, which I believe is somewhere between, isn't it around like 20 to 50 microliter, I think. So this is really nice for my groups that like to sort their samples, because a lot of them will sort directly into our sample buffer, and then we can load directly from sorting. And there's a lot less loss of sample because we need to skin things down and concentrate them. It is also, like I said, much more gentle on the cells, so microfluidics can be quite harsh on cells, especially fragile cell types like granulocytes. It causes a lot of that kind of shear stress to force them through that, as well as the oil itself can sometimes be irritating or activating. So our cells there's no oil anywhere in the system either. So as far as sample preparation, it's going to look very similar to any single cell experiment. You're going to want a nice single cell suspension with high viability, and you know, a nice enough suspension that you have a few multiplets. I think I have groups that get away with loading lower viability samples because we're able to wash away more of that debris because of the open well system. It's not necessarily recommended to run a sample with low viability, but for some of my groups working with patient samples, it's kind of a once in a lifetime sample and you want to be able to get something from it. But that's something that our team will also work with you to talk about your sample preparation techniques and what we can do to optimize that.
I would say there's a bit of a cost benefit as well,
Mhm.
probably 25 to 30% with just list pricing.
Yeah, cheaper.
Cheaper, yeah, and plus if you do decide to do some multiple or multiplexing, because we can load more, you can get more per lane. Another thing to do out there are labs that are close together, and you guys collaborate, or maybe just even are good friends. Those options for that pilot project, that's an 8-lane project, it's an 8-lane for that 5K, for example. So
Yeah, you
can
could
split
split
things
it
up, depending on how that works, so just an option.
How long can you save your cash?
How long can you save the cartridge? 1 year.
How do you prevent more than one cell entering into the single well?
So this has to do with how you prepare that single cell suspension. So we want to make sure that if you're working from a tissue, for example, that it's well digested and that we've run it through some filters to break up clumps. There will be multiplets. Every platform deals with multiplets. But our scanner actually has a predicted multiplet calculator on it based on the counting that you do. So if it looks like it's going to be a bit high, we can filter it again. But we prevent multiplets with the concentration of cells that we load.
So if we get cells consult, the user will set some parameters with single cell already. So
That's right.
we only need to adjust the cell concentration to make sure there's only one cell.
Yes. Yeah.